going to be describing how to do the results section of a scientific report. Again, it's very important that you're following along with your marking criteria or marking rubric to ensure that you understand what each of these elements are. Firstly, we start at how to avoid a fail. In order to avoid a fail, you need to address each of the following key elements. Now, for the results section, there's 3 key elements. Firstly is that you need to include a table and a figure of your key findings. So here we've got an example of a table. We've got some key findings in there, another table, and then there's a figure. So we're showing these results or these findings graphically in some way. Second thing we need to do is include text that describes, or at the very least states, what your key findings are. So again, here we've got some text. So it's not good enough to just have a table and figure. You need to actually say what your results are in text. And we're talking in a paragraph format like this. Now, as we move up to the credit and pass section, firstly, we need to consistently meet the APA Scientific Style Guide for the presentation of tables and figures. And again, that means that we need to cite our tables and our figures in text. So it's no good just having them there, and somewhere within the text there needs to be a reference to it. See, we can see table 1 and 2 here. So see figure 4 here. Also, the tables are labelled above like so with the bold and the small case, and figures labelled below. So tables above, figures below, both cited within text. Need to do this 100% in this section in order to pass into that distinction level. Secondly is when you do your figure, in some way, number 1, you've used the correct data in this figure. Number 2, you've picked the most appropriate figure. And sometimes, there's many correct answers, so you won't get penalised if your figure correctly represents your data and it's showing what it's meant to show, particularly if there's more than one way. But if you've got a picture or a graph that is completely misused and does not show what you're trying to show it, that wouldn't pass this section. Number 2, in the axes, it's labelled correctly. You've got the correct numbers on the axes. Essentially, that the data being represented is the true data. It's not being misrepresented in any way. The next thing you need to do in order to push this up into the distinction level is you need to create a figure that demonstrates your secondary findings. Now, this could be either 2 figures, or this could be a figure with both things on it. In this example, we've got both things on it. So the findings in this was that as the number of bits increases, reaction time also increases. And that's this bottom line we can see represented with the high stimulus response compatibility example. The secondary findings are that with low stimulus response compatibility, the actual reaction time slows down at a more rapid rate, also starts higher, so you're slower with a low stimulus response compatibility model. Keeping in mind that if you are going to present it on the same figure like this, use different symbols or some way to identify the differences between your two different sets of data, your primary or your first major finding, and also your second findings. Third, the next thing in this distinction box, probably the most difficult. It says you need to comprehensively have a comprehensive quantitative analysis of your results, including maybe some percentages or some actual figures. For example, it says here specifically an increase of 1 bit resulted in an average of 23.5%. Now, this is not something that you're going to already have from your lab books necessarily. It's going to require some form of calculation. You actually have gone in here and said, all right, what's the difference between this and this and this and this? So 0 to 1 and 1 to 2. I have to work out some numbers. It's going to be different depending on what situation I'm in. But in some way, I'm trying to quantify using some kind of numerical data what the changes in my experiment was. It could be catching a ball. I caught it 10% more times under certain conditions and so on. Some kind of number changes are very important in this section. And finally, and it's not an example I can show you in this particular one because this lab report didn't do it, but some kind of quantitative comparison to scientific research using a table or figure. So the difference here is we're not attempting to explain the difference, but I might have another line on here, maybe a dotted line that says, example of research average, or, example, this is a line that illustrates some other data from the literature to show some kind of baseline. So my results better than usual, worse than usual. This is going to be hard to do in many cases, but something that compares your findings in this representation, this graphical representation, to some kind of other research findings.